Welcome to the AtlantECO podcast, where we talk about the science behind the AtlantECO project, the Atlantic Ocean, and the human adventures experienced along the way. Here, we have conversations with guests from around the world who work together so that we can better understand, manage, and protect the ocean. So let's embark on the journey of AtlantECO and discover the world that lies above and beneath the surface of the beautiful Atlantic Ocean. Welcome back to the AtlantECO podcast, and we are back with an episode dedicated to discovering a region of the Atlantic with one of the AtlantECO scientists who embarked on the Tara schooner during the Mission Microbiomes of the Tara Ocean Foundation and one of AtlantECO's flagship expeditions. Our guest today is Samuel Chaffron, and he was chief scientist on Tara to study the upwelling in Senegal and Gambia, and he will tell us about the scientific questions that the team were trying to answer. But first, let's get to know our guest. Sam, hi and welcome. It is a pleasure to have this chat with you today.
Hi, Eloise. Well, it's reciprocal. Thanks a lot for the invitation. It's a pleasure to be here with you today as well.
Yeah, and so as I mentioned, we'd like to get to know you a little bit. So can you share with us how your connection with the ocean started, how it evolved, and also tell us about the journey that has led you to where you are today?
Well, actually, I have relatively close connection to the ocean because I grew up about 100 meters from the ocean, the Atlantic Ocean, actually, in Brittany, in Loch Tuddy. So I grew up next to the ocean. My grandfather were fishermen, so I was really close to the ocean, and the ocean has been feeding me for all these years, actually, until I moved out and went on to studies at universities, so mainly in France. A key moment, I think, during these studies has been a scientific internship. The first one I did, it was in Roscoff, in fact, at the marine station in Roscoff, and where I worked on cyanobacteria. That's where it all started, actually, where I started to discover and realize the power of plankton, basically, and especially phytoplankton, and these small cyanobacteria, in particular Prochlorococcus and Synechococcus. And also there, actually, I was involved in building a system to do light and dark cycle to culture the cyanobacteria. And that's where also I started to have first contact with computer science, because we had to control by computer the system. And probably this was also influential in guiding my research or my path towards also bioinformatics, computer science, because I'm also a bioinformatician. And then I moved. I did a PhD in Zurich, in Switzerland, computational biology, systems biology. So trying to understand by modeling microbial ecosystems. And then I moved to Brussels in a bioinformatics lab. I started to work with Tara back in 2011, and also this brought me back to meet again people from Roscoff who were involved in Tara. So yeah, that was it was very pleasant to be able to work with these people again.
Yeah, a lot of stories from people to say, it started because the ocean was right there in front of me. So today we're discussing the leg of Mission Microbiomes, which was focusing on the Senegalese upwelling. So can you explain what the particularities of that region are, and what sort of scientific questions you were trying to answer whilst you were there?
The leg started in Gambia, from Banjul in Gambia, from the capital of Gambia, to Dakar in Senegal. And we sampled this area in front of the coast of Senegal, which is a very productive area. So these waters are very rich, in fact. We call this region an upwelling region because we have in winter enrichment by nutrients that comes from cold water, deep cold water that come to the surface. So here the interest was to better understand this upwelling region, and also in particular the state of this region before the upwelling season, because the upwelling takes place in winter, usually when you have strong wind chasing the hot water in surface. So then this cold water filled with nutrients can come up. and help to create these blooms of plankton, which supports large stocks of fish, actually. This area is a world famous area for fisheries. It's a very productive area. So the idea was to better characterize the pre-upwelling state of this region, because we went there in August, so in summer, so that's not the actual upwelling season, but so we went to sample specific stations that have been sampled regularly by the IRD crews who's working there every year. So the idea was to sample and to try to characterize the pre-upwelling state, because these crews, IRD crews, will actually sample at the same station as us in December. And they will also integrate all the TARA OMICS protocol. So we will be able to compare this pre-upwelling state of the system of the coast of Senegal with the actual upwelling state. So that was the first objective, characterizing the upwelling system there of the coast of Senegal. Another objective was to characterize the oxygen minimum zone that we can encounter there of the coast of Senegal. So this oxygen minimum zone can be found in several regions of the ocean, especially in the Indian Ocean, in the Pacific Ocean also, of the coast of South America and the US, north of the US. So these zones are where we encounter a minimum of oxygen, and this is caused by several factors, which are both abiotic and biotic, so physical processes, but also biological processes. So we still don't fully understand how they form, but we know that they are expanding and that they are modifying the composition of the plankton. They also may have a strong impact modifying the plankton communities on the fisheries, in fact, that are there in this productive area. This second objective was to try to characterize this oxygen minimum zone, which we could sample. We detected it at around 100 meters depth, so we could sample it as well. So that was a successful leg in a way, because we could sample it. Yeah.
Yeah, you set out to do both things, and you managed to sample both environments. So yeah, it'll be interesting to see what results come out of there, and nice to see that there's also collaboration with ongoing, expeditions that already exist, and sharing the data between different projects, different initiatives, so that we can all together work to better understand what is out there and how it functions and how it influences the ecosystem and all the services that these ecosystems provide. So now that we know what you wanted to study, can you tell us how this was studied? So what did you have to do? What will happen next? And also, how will this help to improve our knowledge on how the ocean is doing and how it might evolve over time?
Since this leg was, in fact, also collaboration with this IRD, so the Institut for the Recherche and Development, I don't know how to translate it, but we targeted 5 stations of the coast of Senegal that they are regularly sampling. So we went to sample 2 surface stations, so quite close to the coast, about a few tens of miles of the coast. We sampled only surface. Well, usual protocols were applied. So we used the plankton nets and the pump to sample all the size fractions of the plankton. So from viruses to bacteria, archaea, protists, and metazoan. So two stations close to the coast where we observed high productivity, a lot of diatoms, although it was the pre-upwelling season, but they was already very productive in a way. So that was interesting. And then we also sampled 3 other stations further off the coast, so about 40 nautical miles off the coast. And there we could do a fine scale sampling of surface, and also the DCM, the deep chlorophyll maximum, where we encounter a maximum of chlorophyll, as well as this oxygen minimum zone that was around 100 meters. So it was interesting because we could detect a relatively well-defined deep chlorophyll maximum around 50 meters. So there was quite a lot of plankton there as well, but we can imagine that when the IRD crews will sample, they will actually encounter at the same spots, much more biomass, I suppose. And it will be interesting also to see how this OMZ, the oxygen minimum zone, is impacted by this upwelling system. And do we actually also detect an increase of biomass in this oxygen minimum zone, as it should be the case for surface DCM. We expect to see much more biomass and large blooms, likely of diatoms. So it will be interesting to see how this OMZ will be responding also to this upwelling, to this enrichment.
Yeah, because I guess if there's no oxygen, then it's more difficult for life to prosper and to grow. So it's kind of a contrasting, you've got an upwelling where biomass is developing, you expect to find more plankton there, but at the same time, these zones, there's no oxygen, so how could they grow? So I guess that's the one of the questions.
Yeah, that's one of the main questions, exactly. So there is still some oxygen. It's not completely depleted in oxygen, although of course some plankton won't be able to grow because there is not enough oxygen, especially for all of them. So there should be less biomass. There will be also different species, in fact, developing there, especially planktonic species that are adapted to low oxygen concentration. We know that some bacteria can thrive in these low oxygen concentration zones. So that's why these samples will be very interesting to be compared between the pre-upwelling state and the upwelling state. Yeah, a lot of new insight, I think, will come out of these samples. And so for our leg, the samples are already well shipped to Genoscope, but we will have to wait for the samples of the IRD crews also to be able to compare, right? So this will still take some time, of course, but that's going to be very interesting to be able to compare these two seasons.
Yeah, because one thing is to take the sample, the other is to analyze. There's genomic studies, so you need to wait for all the sequencing to be done. There's imaging studies, and then you get all this kind of data, and then somebody has to look at all this data and make sense of it all. So yeah, we're still a way off of having some form of understanding of what's going on, and maybe we'll come back to you to see what you found out. Maybe in a couple of years' time, we'll see how that goes.
Yeah, it will take some time. It's true that we need some time to sequence, image all these samples, and then what takes even longer is actually to integrate all this heterogeneous data we have. And to interpret what we observe, to mine all this data. So I think that's what takes even the longest time, basically. Yeah. So we have some work ahead.
And we'll come back to it. Maybe, as I said, a couple of years' time. Hopefully, we'll have made good progress there. Turning now to a slightly different subject, but something that we cover in each episode. So with each guest of the expedition, that we've heard that each scientific study was dedicated to a woman who contributed to the field of marine sciences or to the field of marine sports, for example. And in this case, it is actually a little bit different, as we want to have a more general statement on this subject. So can you tell us a little bit about the idea?
Well, yeah, I think it's important to recognize and acknowledge all the work by actually all the women that have been involved in building, making this expedition and this project possible. So I think it's a good idea to acknowledge and say thank you to all the women that have been involved, including you, Eloise, in this project. And all the women, on land, but also at sea. I think I'm thinking about all the women from the Tara Foundation also. There are a number who make this expedition possible. It's maybe hard to imagine, but it's a lot of work to organize all the logistics of this type of expedition, all the communication also, and the outreach. There are many women involved, and for the science, there are also many women involved, actually. It's good to make maybe a general acknowledgement about all the work from these women. And I'm also thinking about the women at sea who have been sailing on Tara, and also the other boats that were involved during the missions.
So as to all the women that made that expedition possible, and then that will in the future also help with the data we mentioned, the amounts of data that has been collected, and that will allow the field to grow and to improve. So
Yeah, of course, there are women in the labs also helping us to make sense of all this data. In our lab, actually, we have PhD students working with this type of data. So at all levels, we have women, right, making this possible. So
and we need them.
And we need them, of course.
We need each other to make to make everything go forward. Okay, well, let's have our conversation go back to your experience on Tara, and to finish on that note, actually, can you tell us what it was like for you? I mean, you mentioned that you've been working with Tara for a while now, but let's focus on this specific experience there. And can you tell us about your life on board, the memories you've brought back from that region, and of course the people? As we said, people are important, so the people that you shared that journey with.
Yeah, sure. Well, actually, that was my my third time on a leg on board Tara, and it's always incredible memories you bring back from this experience. Always memorable, really. I mean, the first time was back in 2013 in Atlantic as well, at the beginning of the Polar Circle expedition from Brittany to Norway. So that was a fantastic experience. And more recently, I embarked twice during the AtlantECO mission. What was very special about this last leg is the fact that I embarked again with two Brazilian scientists, Erica and Pedro. It was very, very nice to be able to embark with them again, and because we had been a great team, actually, working together. And it was also really nice to be able to meet again some of the sailors on board. It takes a few days, but then there is always like a synergy, symbiosis forming on the boat usually, because we need to work hard, right, to make it successful. So yeah, I like this, that somehow we self-organize ourselves on board, and usually it goes always very well, and there are strong bonds, strong connections always forming on during one leg. We also had the chance to observe a lot of sea life, a lot of dolphins, even a turtle. So that was quite nice memories. And one night we even saw a large cruise of dolphins. This shows actually to what extent this region is very productive and rich. You see a lot of life, much more than what we have been observing off the coast of Brazil, actually, during the leg around the sea months, where the region was very oligotrophic, so we didn't observe almost no life, except some plankton, of course, no mammals, basically. And during this leg, we saw a lot of dolphins, and we even saw dolphins playing around the boat in bioluminescent plankton. So that was a very nice memory, actually. Yeah. And we could share that with all the scientists on board and some of the sailors, so it was a very good moment. Yeah.
Yeah, those memories are the kind that will last for a lifetime, for sure.
Exactly. That's true.
So, Sam, I have to thank you for taking the time to speak to us today and to explain, what you were doing while studying the ocean off the coast of Senegal and Gambia. It's been really interesting, and I'm sure we'll be back to hear from the results, if not all of them, maybe initial thoughts and initial results from this expedition and what has been found out. But for now, thank you so much, and let's keep the work going.
Well, thanks to you, Eloise, and it was a pleasure to exchange with you too.
We hope that you've enjoyed today's episode and look forward to seeing you next time. You can follow the AtlantECO project on our website on www.atlanteco.eu, and you can also find us on Twitter, Instagram and Facebook. All the links and information on the project and on today's episode is in the show notes. AtlantECO is a project funded under Horizon 2020, a European Union research and innovation programme.